to go from correlation, because most of what we do, certainly in cognitive neuroscience, most of what we do is correlation. We still don't have very efficient tools to move from correlation to causation. And you can also, for example, search for random mutations in, for example, in mice or in other genetic organisms. And in fact, it may well be possible that among the, the thousands of alleles of different mice, there may be already some that are sort of unconscious, that are like zombie mice, in a sense that have complex behavior, but not those behaviors that we think are associated with consciousness. And also what's important from a theoretical point of view, you want to develop a theory of consciousness. You want to develop a theoretical, probably information-based theory, and Giulio Tononi is doing that together with sort of Edelman, to have a more justified understanding which systems under which conditions show consciousness, based on which criteria. What tools do we need to understand consciousness? What's absolutely essential, and Francis, of course, always emphasizes, we need to acquire much more, particularly if you care about primates, humans and macaques, or other non-human primates, we really need much better and faster methods to acquire neuroanatomical data, wiring data. It's really essential to know, I'm not just recording V1, but I'm recording from a layer 6 cell that projects back to the LGN, or layer 5 cell that projects back to the pulvinar, and I want to have a tool that lights up all those connections so I can then inactivate them or record from them. We need those tools. We need to develop electrical tools and optical tools to record not just from. So in humans, we can now record from 50 neurons, and in rodents, you can do it from 200 using tetrodes, but you really want to have tools where you can sample 10,000 neurons, the activity at the millisecond time scale. Then we need the mathematics. This hasn't really been mentioned at all. We need to understand how very complicated heterogeneous networks, so all the neural networks that people have done, like Hopfield and other people, they're really very simple neural networks. They're all very homogeneous, and you have either things that excite or inhibit. Neurons are very heterogeneous. They spike, they burst, they have different neurotransmitters, they have different dendritic patterns. They have calcium-based spikes, they have sodium-based spikes. And so we need a mathematics. What happens if we have 10,000 neurons, or 100,000 neurons, or 10 billion neurons, very complicated ways, and interacting at the millisecond time scale, and we don't really have such a mathematics right now. We need, this has been mentioned before, we don't have this at all in humans. We want to be able non-invasively to sample the human brain activity at the millisecond time scale, millisecond and micrometer. We want to do it both for clinical purposes, because we want to help people who are paralyzed or locked-in syndrome without opening the skulls, and we also want to do it for scientific reasons. And right now, this is really a huge hole in the spectrum of our technologies that we have. And again, we need ways to perturb and intervene in the mammalian CNS in a very delicate and transient manner. I found this great quote in a review that Bateson wrote during the First World War, when he reviewed Thomas and Morgan's book, The Mechanism of Mendelian Heredity. So Morgan, at the time still at Columbia, I believe, made the point that genetic information is stored along one-dimensional strings in modern language. And at the time, people didn't understand, even people who are materialists like Bateson didn't understand how chemistry could be used to store all the prodigious amount of information that makes up an individual and pass it on to the descendant. People thought they understood chemistry, but they didn't know how this could work. And so people like Bergson developed, new philosophy. We need an élan vital, right, vitalism. Or people like Schrödinger, as late as 1944, said we need new physics in order to explain this. And so he has this quote, the properties of living, well, you can read it. So the problem here was people didn't even, at this time, they didn't even have the idea of a specific macromolecule. They didn't realize that hemoglobin in me is not just a colloidal, it's not just a collection of different molecules that have a huge spectrum of molecular weight. No, but it's all one and the same molecule, replicated, a gazillion times. They didn't understand the prodigious power of strings of nucleic acids. And so when, I go a lot to these consciousness meetings, and people constantly talk about quantum gravity and quantum computation and, and sort of other fancy, spooky stuff. And we really haven't exploited the power of the brain. It's by far the most complex system, and we don't know it. And we really have no idea how we just don't understand the power of the brain yet. And so we shouldn't sort of call up for all sorts of fancy new laws before we sort of really understand the complexity of these biological networks. And so I'd like to leave you with this last point, which is Francis and my entire emphasis, that consciousness should be treated as an empirical problem to be tackled by the biological sciences. Thank you.
Thank you. He will be back when he comes up here. Next, we're going to turn to someone whose questions about consciousness may be coming from a different angle. Dr. Patricia Smith Churchland is a Canadian-American philosopher. Currently, she is chair of the University of California, San Diego Philosophy Department, an adjunct professor at the Salk Institute for Biological Research, an associate of the Computational Neuroscience Laboratory at Salk. She is surrounded by philosophers, as her husband, Paul Churchland, also pleads guilty to that profession. Her recent work focuses on neuroethics, the attempt to understand choice, responsibility, and the basic of moral norms in terms of brain function, brain evolution, and brain culture interactions. Please welcome Dr. Churchland.
I'm not sure. Oh, yes, I am wired. Great. Has anybody got any glutamate left? It's very late in the day, and it's been a very, very rich and really very wonderful day. Although my brief is to talk about consciousness, I'm actually going to say very little about it. What I would really rather do is to sort of take a very broad picture about some of the open questions in neuroscience, and just to say a little bit about what we don't know, given the background of really the stupendous discoveries that have been made in neuroscience over the last couple of decades. I begin with this particular slide really just to remind ourselves that our brains are as they are because they are the products of natural selection. And as we now know, the conservation of structures is really very remarkable across species. And it's unlikely that we are going to find in ourselves structures and even functions that are absolutely unique and have no precursors in other animals. All right. The other point that's useful to make from the background of evolutionary biology is this, that we differ from plants, that is, animals differ from plants, fundamentally in having a nervous system that enables us to move. We don't have to wait and take life as it comes. We can look for food, look for mates, see to it that our offspring are nurtured, and so forth. Motor control, or more broadly perhaps, behavior, is really the fundamental thing for which nervous systems are designed. And I think it sometimes is forgotten as we explore the nervous system from its sensory aspects or from the aspects of learning and memory, that whatever those functions are in their nature, they have to be serving behavior and motor control. So that perception is not there for the sheer wonderful beauty of it. It's there only because it serves behavior and motor control in some fundamental way. An organism that has absolutely stupendous color vision, but where that color vision in no way informs its behavior is not going to survive and consequently will not pass on its genes. Oh, let me just go back. I'm sorry. It was really Paul MacLean who put the